Hello, welcome to my channel Deep Learning Biology. My name is Dr. Vikas Mahardwaj, and within this video I am going to teach you structure of DNA. So here we are going to discuss the structure of DNA. Hopefully you have watched my earlier videos in which I have discussed how it was being proved that DNA is the genetic material. Now in this video, you are going to learn the structure of DNA. Now here I just want to tell you, you must be knowing about cell theory. Who proposed the cell theory? Sleeden and Swan. Now as per cell theory, all living organisms, whether plants, whether animals, whether microorganisms, they are made up of cell. Cell is the fundamental unit of life. Now here if you see a cell, within the cell you are going to have nucleus, and within the nucleus you are going to have genetic information. Yes, it is true, in the case of bacterial cell you are not going to see any nucleus. There you are going to see the nucleoid. But here if you see in the eukaryotic cell you are going to have nucleus, and within the nucleus you are going to have genetic information, and this genetic information, it is being stored within the DNA. Try to think here. Within your body, you are going to have millions of cells. And all these cells, they carry genetic information. From where? Of all of your ancestors. Within your body, you are going to have genetic information of all of your ancestors. And here, this information, it is being stored within the DNA. So in today's class, we are going to learn the structure of DNA. Now here, I just want to tell you, hope you remember, in the year 1869, Frederick Mister, he isolated the DNA. In the year 1953, Watson and Crick, Morris Wilkins, based on the work of Rosalind Franklin, all of them, they proposed the structure of DNA. So we have already discussed the history, how the structure of DNA was revealed. Please have a look into that lecture. Now in this video, we are going to discuss the structure of DNA. Now here, the first point I just want to tell you, hopefully you remember, hopefully you know, DNA, it is going to have double helical structure. So if you look into the structure of DNA, it will look like, okay, so if you look the structure of DNA, so it is going to have two helix, okay, and both of the helix, they are going to run in anti-parallel direction. Now here if you see that if you turn this model by 180 degree, it will remain same. So keep in mind, in the case of DNA, you are going to have two chains of polynucleotides. So we are going to discuss what are nucleotides. So keep in mind, here you are going to have two chains of polynucleotides, and both of the chains, they are going to run in anti-parallel direction. So whenever we are going to discuss Watson and Crick model of DNA, keep in mind both of the chains, they are going to run in anti-parallel direction. So here in the case of DNA, you are going to have two chains of polynucleotides, and both of the chains, they are going to run in anti-parallel direction. Now let us discuss what is a nucleotide. What is a nucleotide? Base plus sugar plus phosphate. Keep in mind, base plus sugar plus phosphate is nucleotide, while base plus sugar, it is nucleoside. So hopefully it is clear to you what is nucleotide. Nucleoside plus phosphate, it is nucleotide. So base plus sugar plus phosphate is nucleotide. Base plus sugar is nucleoside. Now here if you see the structure, okay, you can see a base, it is being attached to the deoxyribose sugar. In the case of DNA, always remember the sugar is deoxyribose, means here in the case of sugar at the 2' position you are going to have hydrogen, okay. Now this sugar, it is being attached to the phosphate group. So here this is nucleotide. Now keep in mind, in the case of DNA, the sugar is deoxyribose. Here at the 2' position, you are going to have hydrogen atom. You are not going to have hydroxylic group. Yes, in the case of RNA, keep in mind the sugar is ribose. So you are going to have two types of nucleic acid. One is DNA. In the case of DNA, always remember the sugar is deoxyribose. In case of RNA, the sugar is ribose. In case of DNA, you are going to have at the 2' position hydrogen atom, while in case of RNA, you are going to have OH group. Now here, I just want to tell you, if you see the base, base is actually a flat structure. And here, I also want to tell you, base, they are hydrophobic. Hopefully you remember the structure of DNA. Within the DNA, the base, they are inside. So here we are going to discuss that whenever something is hydrophobic and if surrounding environment is hydrophilic, most of the times hydrophobic things, they will be inside. So same thing is going to happen in the case of DNA also. Now focus here about the base. In the case of DNA, you are going to have two kinds of base. One is purines, another is pyrimidines. So here purines, generally they are being found are adenine and guanine. In case of DNA, you are going to have adenine and guanine, okay, as the purines. While if you see thymine and cytosine, they are pyrimidines. So these four base, they are being found generally in the case of DNA. So you can say that within the DNA, you are going to have four letters, A, T, G, and C. Keep in mind purines, adenine and guanine, okay, and pyrimidines are thymine and cytosine. Now here in the case of RNA, you are going to have uracil in place of thymine. Always remember in RNA, the sugar is ribose. And another important point, in the case of RNA, you are going to have uracil in place of thymine. So in RNA, you are going to see adenine, guanine, cytosine, and uracil. So hopefully all these points are clear to you. Next here, I just want to tell you, you must remember some of the things related to purines. Now here, I just want to tell you, in the case of purines, what you are going to see, you are going to see double ring. So it is a 9-member ring. Okay, so 9-member ring it is there. Okay, so here you can see in the case of adenine, okay, what you are going to see, you are not going to see any oxygen atom. Always remember, you can call adenine as 6-aminopurine. If you remember this way, you can easily draw the structure of purine. So here you can remember adenine as 6-aminopurine. And keep in mind, in the case of adenine, you are not going to see any oxygen atom. Many times in various competitive exams, question is being asked, which base is not going to have oxygen atom? So keep in mind, the right answer is adenine. Now here, if you see the structure of guanine, okay, here in the case of guanine, you are also going to see the double ring, okay. Now here, you can remember the structure of guanine, if you can remember this as 2-amino-6-oxopurine. Okay, 2-amino-6-oxopurine. So here at the second position, you are going to have amino group. At the 6th position, you are going to have the oxo group. So 2-amino-6-oxopurine is guanine, 6-aminopurine is adenine. Now here, if you see the case of pyrimidine, in the case of pyrimidine, what you are going to see, you are going to see single ring. Here, if you see the structure, so this is the structure of thymine. Now here, if you see the chemical structure of thymine, it is 2,4-dioxo, 5-methyl pyrimidine. Okay, so here at the second position and fourth position, what you are going to see the oxygen atom attached. Okay, so 2,4-dioxo. At the 5th position, you are going to have methyl group, 2,4-dioxo, 5-methyl pyrimidine. Same way, if you see the structure of cytosine, here also you are going to see the single ring. Okay, so here, if you see the chemical name of the cytosine, it is 2-oxo, 4-aminopyrimidine. So hopefully it is clear to you all the structure of different bases which are being found in the case of DNA. Now here, hopefully you know, I have told you, in the case of DNA you are going to have thymine, while in the case of RNA you are going to have uracil. Now just look the difference between the structure of thymine and uracil. So here if you see the chemical structure of thymine, okay, so here the chemical name is 2,4-dioxo-5-methylpyrimidine. Okay, the chemical name of thymine is 2,4-dioxo-5-methylpyrimidine. While if you see the case of uracil, here if you see the chemical name is 2,4-dioxopyrimidine. Now try to see the difference between the chemical structure of thymine and chemical structure of uracil. Did you notice the difference? In the case of thymine, you are going to have 5-methyl, while in the case of uracil, what is going to happen? The 5-methyl group is absent. So here, if you look clearly, you can say thymine, it is actually the methylated form of uracil. Okay, thymine is methylated form of uracil. Keep in mind. So there is one difference of methyl group. So hopefully you know thymine is generally found in the case of DNA, and uracil it is generally found in the case of RNA. So keep in mind that in the thymine is methylated form of uracil. Now let's move further. Here I just want to tell you, if you see, most of the times whenever we are going to read any DNA sequence, the general rule is that we are going to read it in 5' to 3' direction. Okay. Now here a question arises, why we are going to use these words prime. Okay, we are going to discuss this point, why we are going to use this prime. Okay. Now here if you see the nucleotide, okay, so here in the case of nucleotide, what is going to happen? Base, it is being attached to sugar, and sugar is being attached to phosphate. Now try to think here. Base, they are going to have atoms, and these atoms they are going to have some numbering 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. Same way the sugar is also going to have carbon atoms. This is also going to have some numbering. Okay, 1, 2, 3, 4, 5. Now here to distinguish the atoms which are present on sugar, the atoms which are present on base. So generally the atoms which are present on the sugar, they have been given the prime symbol. Hopefully it is clear to you why we are going to use the term prime. It is simple to distinguish carbon atom which are present on base and carbon atom which are present on the sugar. Now here I just want to tell you, you must be knowing the complementary nature of DNA. DNA is going to have complementary nature. Means if you are going to write one sequence of DNA, let's suppose it is 5' A, T, G, C, T, A, 3'. Okay, you can easily write the other strand. Okay, just you have to follow the rule. What is the rule? A is going to pair with T, okay, and G is going to pair with C. In front of A, you are going to write T, and in front of G, you are going to write C. So this is the complementary nature of DNA. Always remember, in case of DNA, adenine, it is going to pair with thymine, and there are 2 hydrogen bonds. Guanine, it is going to pair with cytosine, and there are 3 hydrogen bonds. Now here, keep in mind, at the 5' end, you are going to have the phosphate group. At the 3' end, you are going to have the OH group. Same is in the case of the other side also. At the 5' end, you are going to have the phosphate group. At the 3' end, you are going to have the OH group. So hopefully it is clear to you that the DNA, it is going to have complementary nature. So most of the times questions are being asked, okay, like this way, that if you are going to have a double-stranded DNA which is going to have 20% adenine, okay, what will be the base composition of other bases? So it is quite simple. If you are going to have 20% adenine, means you are going to have 20% thymine in that double-stranded DNA. Same way, if you see adenine plus thymine, it will be 40%. Rest of the bases will be 100 minus 40 means 60%. So guanine and cytosine will be 60%. Just you do the half, because amount of guanine will be equal to amount of cytosine. You can easily tell each base's amount. So hopefully you have understood this point clearly. Now here I just want to tell you, you must be knowing about the Chargaff rule. What Chargaff rule says, here I have already told you that in case of DNA, adenine, it is going to pair with thymine, and guanine, it is going to pair with cytosine. So here keep in mind that in the case of DNA, there is a pairing between purines and pyrimidines. So adenine and guanine, they are purines. Thymine and cytosine, they are pyrimidines. Here I also want to tell you that this is not going to represent the number of hydrogen bonds. Here keep in mind, between adenine and thymine, you are going to have 2 hydrogen bonds. Between guanine and cytosine, you are going to have 3 hydrogen bonds. Now here, in the case of double-stranded DNA, amount of purine, if you sum adenine plus guanine, it is going to be equal to thymine and cytosine. So amount of purines is equal to amount of pyrimidine in the case of DNA, and this is known as Chargaff rule. Now here, if you see in the case of DNA, what is going to happen? The base, it is being attached to sugar, and sugar is being attached to the phosphate group. Now here you look again the nucleotide. Okay, here the nucleotide base, it is being attached to the sugar, and sugar is being attached to the phosphate. Now keep in mind, in the case of purines, in the case of purine, nitrogen 9, okay, 9th number atom, nitrogen, it is attached to the sugar, okay, and this is a glycosidic linkage. So N9 of purines, it is attached to carbon 1 of the sugar. So this you keep in mind. Same way, in the case of pyrimidine, what you are going to have, nitrogen 1, it is being attached to carbon 1. Okay, so base, it is being attached to sugar. So here in the case of pyrimidine, it is the N1, it is being attached to carbon 1. While in the case of purines, nitrogen 9, it is being attached to carbon 1. So here you can see the structure of purine, and here you can see the structure of pyrimidine, how purine, it is being attached to the sugar, and how pyrimidine, it is being attached to the sugar. Now here I just want to tell you, we are going to discuss the structure of DNA, but here I also want to tell you some important point related to structure of proteins. Hopefully you know, in the case of proteins, in the case of cellular proteins, the proteins which are present within the cytoplasm, okay, not the membranous proteins. So in the case of proteins which are generally found within the cytoplasm, what is going to happen? The outside environment, it is hydrophilic. Within the cytoplasm, you are going to have water. So you are going to have 70 to 75% water content. So if you are going to have a cytosolic protein, always remember hydrophilic amino acid, they are present outside. Hopefully you know proteins, they are polymer of amino acids. So always remember hydrophilic amino acids, they are present outside, while the hydrophobic one, they are present inside. So hopefully you know that hydrophilic things, they actually love water. Okay, here the amino acids which are hydrophilic, they will love to be outside, while the hydrophobic things, they actually have a fear from water, they try to be inside. So keep in mind, in the case of proteins and only in the case of cytosolic proteins, what you are going to see, hydrophilic amino acids, they are present outside, hydrophobic amino acids, they are present inside. Now here keep in mind this concept, because the same concept we are going to apply in the case of DNA also. Now here in the case of DNA, keep in mind the base, they are hydrophobic. In the case of DNA, the base, they are hydrophobic, and that's why they are inside. While if you see the sugar phosphate backbone, okay, if you see the DNA, you are going to have a sugar phosphate backbone. The sugar phosphate backbone is hydrophilic, and that is outside. So same thing as like proteins, okay, the hydrophobic things, they are inside, while the hydrophilic things, they are outside. Now here I just want to tell you, here you can see the structure of DNA. So here you can see the base they are present inside, while if you see the sugar phosphate backbone, it is present outside. Hopefully this point is clear to you. Next here I just want to tell you about phosphodiester linkage, because in case of DNA what you are going to have, phosphodiester linkage. So here you can see in the DNA you are going to have a chain, okay, where you are going to see the phosphodiester linkage, and I have told you the sugar phosphate backbone, it is outside. Within the sugar phosphate backbone you are going to see the phosphodiester linkage. Let's have a clarity, what is this phosphodiester linkage? Now here in the case of phosphodiester bond, what is going to happen, phosphoric acid, it is linked to two alcohols. So phosphoric acid, it is linked to two alcohols. Now try to see here. Here what is going to happen, in the case of nucleotide base, it is being attached to sugar. So there is a glycosidic bond between base and the sugar. Next, the sugar, it is being attached through a phosphate group to another sugar. So on sugar you are going to have alcoholic group, on another sugar you are also going to have alcoholic group, and here you are going to have the phosphate group. So here what is going to happen, two ester group are being formed. So this is phosphodiester linkage. So hopefully it is clear to you what is phosphodiester linkage. Next here I just want to tell you, you can represent the DNA structure by a shorthand representation. It is very simple. You can draw line this way. Here I have shown you the phosphate group, and hopefully you remember the phosphate group, it is going to start from the 5' position. At the 3' position you are going to have the OH group. So here we are going to write the base, okay. So here we are going to represent the base, okay. So here within the DNA what is going to happen, base, it is being attached to the sugar. So here you are going to have the sugar, you can represent here sugar molecules, and the base is being attached to sugar, and sugar it is being attached to the phosphate, and in between you are going to have the phosphodiester linkage. Always remember at the 5' end of DNA you are going to have the phosphate group, and at the 3' end you are going to have the OH group. So hopefully it is clear to you how you can represent the shorthand notation of DNA. So here we have shown only the one strand. Now here I just want to tell you, a question may arise in your mind, what actually stabilizes DNA? Now here if you see, in case of DNA you are going to have covalent bonds, okay. In the chains you are going to have atoms being joined through covalent bonds. Now you must be knowing, generally the covalent bonds, they are being formed by sharing of electrons. So here in the case of carbon you can have one covalent bond. In carbon you can have double covalent bond, you can have triple covalent bonds. It is based on the number of electrons being shared. Now here in the case of DNA, what is one of the most important thing you are going to see, that you are going to see hydrogen bonds between adenine and thymine and guanine and cytosine. Now here I just want to make you clear what are actually hydrogen bonds. So try to see here, let's suppose you are going to have a water molecule. So in the case of water molecules, between oxygen and hydrogen there is covalent bond, there is sharing of electron, but oxygen is more electronegative. What will it do? It will pull the electron towards itself. So here oxygen is going to carry negative charge and hydrogen is going to have positive charge. So in this situation, if there is another water molecule which is also going to have the same thing, oxygen is going to carry negative charge and hydrogen is going to have positive charge. Now always remember, in different charges, in between different charges you are going to have attraction. So here if you are going to have attraction between positive hydrogen and negative oxygen, what you are going to see, you are going to see the formation of hydrogen bonds. Yes, these bonds are weak, okay, they are not strong, but still within a molecule, if you are going to have more number of hydrogen bonds, what is going to happen? The molecule will be stabilized due to the presence of hydrogen bonds. So here keep in mind, in between adenine and thymine, you are going to have 2 hydrogen bonds. Between guanine and cytosine, you are going to have 3 hydrogen bonds. And these hydrogen bonds, they gave stability to the DNA. Now here you can see the structure. So here at the 5' end, you are going to have phosphate group. At the 3' end, you are going to have OH group. Between adenine and thymine, you are going to have 2 hydrogen bonds. Between guanine and cytosine, you are going to have 3 hydrogen bonds. These hydrogen bonds, they are going to give stability to the DNA. Now, hopefully you must be knowing that if you heat the DNA, you can separate both strands of DNA. Okay, you can separate both chains of DNA. Just if you heat it, what will happen? The hydrogen bonds between DNA, they will get disrupted. So keep in mind, in the case of DNA, if you heat, what will happen? The 2 strands of DNA, they will separate. But if you allow it to cool, both strands of DNA, they will reanneal. Now keep in mind, in between A and T, you are going to have 2 hydrogen bonds, and between G and C, you are going to have 3 hydrogen bonds. Now we are going to discuss some details related to the structure of DNA. First thing is, keep in mind the diameter of DNA is 20 angstrom. 20 angstrom, you can represent it is equal to 2 nanometer. So if you see, I have told you DNA is double helical. So when you see the diameter is 20 angstrom. So here, everything, it is being fitted in this small diameter. Now here, I have told you there is hydrogen bonding between A and T, and there is hydrogen bonding between G and C. So adenine, thymine, they are present in this space. Guanine and cytosine, they are present within this space. In the case of DNA, another factor which is going to stabilize it is the base stacking interaction. I have told you hydrogen bonds, they are going to give stability to the DNA. Same way here, what you are going to have, base stacking interaction. I have told you that base, they are flat and they are hydrophobic. So when these hydrophobic things, they are being stacked one above other. Okay, so what is going to happen? They are going to be stabilized by this pi-pi electron clouds interactions. So when hydrophobic base, they are being stacked one above other, so what is going to happen? They also gives stability to the DNA. So here what you are going to have, pi-pi electron cloud interactions, and these interaction actually gives stability to the DNA. So here keep in mind, in the case of DNA, you are going to have base stacking interaction. Next here, I just want to tell you, keep in mind DNA, it is generally found in the form of B form. Okay, so B form of DNA, it is right-handed. When you see the helix, you are going to have 2 types of DNA. You can have 2 types of DNA. One is right-handed double helix, another is left-handed double helix. In the case of right-handed double helix, what is going to happen? If you see the chain from bottom to top, they are moving towards the right hand. In the case of left-handed DNA, what is going to happen? If you see from bottom to top, the chains, they are moving towards the left hand. So keep in mind, you are going to have 2 types of DNA you can have. One is right-handed DNA, another is left-handed DNA. Always keep in mind, B form of DNA, the natural form of DNA, it is right-handed, but keep in mind, there is one example of left-handed DNA is ZDNA. ZDNA is left-handed DNA. Next here, we are going to discuss how many base pairs they are present in 1 ton of DNA. Always keep in mind, in case of DNA, you are going to have 10.5 base pair per ton of DNA. So DNA, it is going to have ladder-like structure. So it is going to have tons. So within 1 ton, you are going to have 10.5 base pair. And sometimes we write 10 base pairs in 1 ton. So keep in mind, in DNA, you are going to have tons, and 1 ton of DNA, you can keep in mind, you are going to have 10 base pair. Now, these 10 base pairs, they are going to have some length. The length of 10 base pair, or you can say 1 ton is 34 angstrom. Okay, you can write this as 3.4 nanometer. Now here, if you see one base pair, it is going to have a length that is equal to 3.4 nanometer divided by 10. It will come out 0.34 nanometer. So here you can see that base, they are flat. Okay, and one base, it is being stacked over another base, and here you can see one base, it is going to have a length that is 0.34 nanometer. Okay, and this is also known as helix rise per base pair. So here, if you see within your genome, within the human genome, you are going to have 3 billion base pairs. 3 billion letters are there. So here, this actually represents the haploid DNA content of human genome. If you want to know what is the diploid DNA content, you just multiply it by 2. So total DNA content, it will be 6 into 10 to the power 9 base pair. Now if you want to calculate what is going to be the length of this DNA, you just multiply it by 0.34 nanometer. Okay, so here you can easily solve what will be the length of DNA. It will come out 2 meter DNA. Now you can easily remember 2 meter DNA, it has been packed within each of your cell. Okay, so in a very small space, in a very small nucleus, you are going to have 2 meter DNA. Now here by this picture, you can remember these parameters. So here I have told you the diameter of DNA is 20 angstrom. Okay, 1 ton of DNA, it is going to have the length that is 3.4 nanometer. Okay, and you can see also the length, okay, of one base pair, or you can see the distance between each base pair is 0.34 nanometer. Now here I just want to tell you that here I have told you the diameter is 20 angstrom, that is 2 nanometer, and 1 ton of DNA, it is going to have 10 base pair. Now 1 ton means one complete 360-degree rotation. So here you are going to have 360-degree rotation, and you are going to have 10 base pair. You can easily calculate how each base pair is going to rotate. Okay, so I have told you that these base pairs, they are being arranged like a ladder. So here they are not just perpendicular, so they are being rotated. So here you are going to easily calculate the rotation, it will come out. Now here you must be knowing, in the case of DNA, you are going to have 2 grooves. One is major groove, another is minor groove. Now here keep in mind, the major groove, it is wider. If you see the width, it is 22 angstrom. So here, if you see in the case of DNA, you are going to have major groove, minor groove, major groove, minor groove. Now here I just want to tell you, major groove, it is going to have a comparatively big width. So here most of the proteins, they can easily fit. Always keep in mind, most of the proteins which are going to interact with the DNA, they are going to interact at the major groove. Next here, keep in mind, in the DNA you are going to have minor groove. Minor groove is narrower. Next important point here, keep in mind, in the case of DNA, the base, they are tilted. Okay, they are not present perpendicular to the helix axis. They are not in perpendicular right angle. So here keep in mind, the base, they are tilted by 6 degree. So here in the case of DNA, the base, yes, I have told you the base, they are present inside, and if you see their position, they are not perfectly perpendicular if you compare with the helix axis. So here keep in mind that base, they are tilted by 6 degree. So here you can see that these base, they are not perpendicular if you have the helix axis. Okay, so here I have shown you that a base, it is present in a perpendicular way, but in actually this is not the situation. If you see the picture here, the base, they are being tilted by 6 degree. Now another important point here, keep in mind, the sugar conformation in DNA. The sugar conformation in DNA. So here you must be knowing the sugar, it is being attached to the base. So there is glycosidic bond formation. In case of purines, you are going to have two conformation. One is syn and anti. So if you see the picture here in the case of base, okay, it will be away, okay, in a anti position. It is away from the sugar, or it will be on the top of the sugar. Okay, so one can be syn position, another can be anti. So here the purines, which are going to have 9-member rings, they can be present in both conformation, syn conformation as well as anti conformation. While if you see the pyrimidines, they are going to have 6-member ring. Only they are going to be present only in anti conformation. So here if you see the structure of the pyrimidines, okay, here what you are going to have, the pyrimidines, they are present, okay, they are linked through glycosidic bond to the sugar, okay, but here you are going to see only the anti conformation. Now here the question arises, why you are going to see only the anti conformation in the case of pyrimidines. Now here if you see in the case of pyrimidines, you are going to have oxygen atom, okay, and if this if it is going to be in syn conformation, what will happen? There will be a repulsion. So here in the case of pyrimidines, keep in mind oxygen atom which is present at the carbon number 2 position, it do not allow the syn conformation. Hopefully every point related to sugar conformation in DNA, it is clear to you. Next important point here we are going to discuss, that is the most important point, okay, the sugar pucker conformation. In case of DNA, in case of B form of DNA, keep in mind the sugar, they are going to have C2 prime endo conformation. What does it mean? Here just look into the atoms which are present within the sugar, okay. Here all these atoms which are present in the sugar, they are not planar, they are not going to lie in a single plane. But here if you see the carbon number 2, it is present inside if you compare it with the position of carbon number 5. So here both are inside. So this conformation, it is known as C2 prime endo. So in case of B form of DNA, you are going to have C2 prime endo conformation of sugar. Yes, other conformation are also there. We are going to discuss that when we are going to discuss the difference between A form of DNA, B form of DNA and Z form of DNA, that we are going to discuss in next video. So hopefully in this video you have learned every basic points related to structure of DNA.